The purpose of this multi-part video is threefold. First, to define some of the nomenclature of tumor biology. Secondly, to describe the differences between benign and malignant tumors. And thirdly, to explain the significance of grading and staging. I will start out by defining some terminology. Now, this is an overview slide of tumor classification that starts with normal structures on the left and ends with cancers on the right. Now, the first term shown in red here is tissue, which you've probably used yourself. It is often used rather loosely in conversation. However, in histology, which is also known as microscopic anatomy, it has a specific definition, which is as follows: a group of cells and the associated extracellular matrix with a similar structure and function. In the human body, there are some 200 or so different types of tissues. The exact number depends on how far you want to parse it. Now, the different types of tissues can be organized into four major tissue types, which are epithelium, nervous, muscle, and connective tissue. I will return to these subdivisions later, since they are important for cancer classification. Now, the term tissue includes both normal structures as well as tumors, which is the next term I would like to define. Originally, the term tumor referred to a swelling of any type, which could be either a normal physiological response, such as inflammation, or to a neoplasm. Currently, the term tumor is used to denote an abnormal growth that is uncoordinated with and exceeds that of normal tissue. So in this regard, the term tumor can be used synonymously with neoplasm, which means new growth. Now, tumor/neoplasm can be either benign or malignant in nature. Benign tumors can be thought of as being mild and generally do not cause problems. They are neither progressive nor recurrent, and potentially reversible. However, they can cause problems indirectly if they press on other organs or release excessive amounts of hormones, such as in the case of an adenoma of the pituitary. On the other hand, malignant tumors do tend to cause harm or death. Indeed, in the United States today, 1 in 5 deaths is due to a malignant tumor, and 90% of this is due to metastases. Now, another name for malignant tumor is cancer. At this point, I would like to consider benign tumors in greater detail. Benign tumors are generally designated by adding the suffix "oma" to the cell of origin. For example, a benign tumor of fibroblast is a fibroma. A benign tumor of cartilage cells is a chondroma, and a benign tumor of glandular epithelium is an adenoma. Now, it is important to note that you should not confuse the suffix of just "oma" with carcinoma or sarcoma, which also contain an OMA, but are associated with malignant tumors. Now, there are several exceptions to this nomenclature, which you will just have to memorize. One of these is melanoma, which is a tumor of melanocytes. You would think from the name that it would be benign, but it is in fact highly malignant. A better name would have been melanocarcinoma, but because of tradition, the name has not changed. Another exception is carcinoma in situ, which from the name you would think would be malignant, but it's not. It is in fact a benign condition. Malignant tumors are classified according to the identity of the primary tumor, not to metastases. So, for example, metastases of breast cancer in the lung is not lung cancer, but rather metastases of breast cancer in the lung. Now, classically, malignant tumors have been divided into 4 major categories based upon the tissue of origin and their clinical features. These 4 are carcinomas, sarcomas, hematopoietic tumors, and neuroectodermal tumors. This next slide is a color-coded scheme showing the source of these different types of tumors. Remember I told you before that there are four major categories of tissues: epithelium, nerve, muscle, and connective tissue. And these are shown in the four quadrants of this slide. In red here are carcinomas, which are derived from epithelial tissue. This is the most common type of cancer and is responsible for the majority, approximately 84%, of cancer-related deaths. Now, carcinomas can be subdivided into two major groups. Firstly, carcinomas proper, which are derived from non-glandular epithelium and includes such things as squamous cell carcinoma of the esophagus. And secondly, adenocarcinomas, which are malignant tumors that arise from glandular epithelium and often retain a glandular growth pattern. An example of this would be prostate adenocarcinoma. Next, shown in blue, are the sarcomas. The prefix sarc means fleshy. These are derived from non-epithelial tissue of mesodermal origins and include muscle and most of connective tissue, with the exception of blood. Now, sarcomas are relatively rare and constitute only about 4% of clinically important tumors. Shown in green here are the hematopoietic tumors that arise from the precursors of the formed elements of the blood, that is, the erythrocytes, the leukocytes, and the platelets. These tumors can be divided into two major groups: leukemias, which tend to primarily involve the bone marrow, and lymphomas, which arise from neoplastic lymphocytes and tend to involve the lymphoid organs, which include the lymph nodes, the spleen, and thymus. And finally, we have the neuroectodermal tumors that are shown in purple. These tumors are set apart from the others because the distinction between benign and malignant is not as clearly defined. For example, gliomas tend to be highly invasive but do not metastasize. However, the location in the central nervous system makes them difficult to remove without causing further damage. These are often lethal tumors, but fortunately they are also relatively rare. This next slide begins the second major topic of this video series, namely the characteristics that distinguish benign from malignant tumors, which are differentiation, growth rate, local invasion, and metastasis. I will start out by considering the first of these, differentiation, which refers to how closely the tumor cells resemble their normal counterparts. In general, benign tumors are well differentiated, while malignant tumors may vary from well differentiated to totally undifferentiated or anaplastic. However, malignant tumors always show some loss of differentiation as compared to their normal and benign counterparts. This slide shows some of the characteristics of benign and malignant neoplasms, with benign tumors on the top and malignant tumors on the bottom. Shown in red here are hyperplasia and dysplasia, both of which are associated with benign tumors. Hyperplasia refers to an excessive number of cells. However, the cells themselves are morphologically normal unless indicated otherwise. Dysplasia, on the other hand, refers to cells that have an abnormal appearance under the microscope, but this abnormality is not as extreme as that of malignant tumors. The histological alterations seen in dysplasia include changes in the nuclear size, shape, or staining, the ratio of the nucleus to the cytoplasm, the staining of the cytoplasm, or alterations in the normal sequence of cell differentiation. Now, both hyperplasia and dysplasia are generally reversible. Examples of these are shown in the next slide. This slide shows several views of ductal epithelium of the mammary gland. Note that the normal duct on the upper left is relatively small, contains a large lumen, and is surrounded by two layers of epithelial cells, as indicated by the location of the nuclei. Note, you cannot generally distinguish the boundary of individual cells by H&E staining as shown here. Now compare this with the hyperplastic condition shown in the upper right side. Please note that the hyperplastic duct is larger and contains multiple lumens, and there is an excessive number of cells here. However, if you compare the morphology of the cells to that of the normal gland, you will note that they are relatively similar in most respects. The nuclei are of a similar size and structure, and there are two layers judging from the nuclei surrounding each of the lumens. Move on now to the sample labeled dysplasia or carcinoma in situ on the lower left side. This figure was taken at a low magnification, so it is actually much larger than the other specimens. Note that it contains a number of spaces that correspond to lumens, but otherwise consists of a solid sheet of cells. When you compare the morphology of the individual cells to its normal counterpart, you should note that it's much more uniform in nature. The carcinoma in situ has lost the normal two-layer arrangement of the normal epithelium. Remember that even though it's termed a carcinoma in situ, it is still a benign condition since the tumor cells have not moved past the basement membrane, as I will describe later. Returning now to the features of benign and malignant neoplasms, you will note that one of the possible characteristics of malignant tumors is anaplasia, which refers to a lack of differentiation. So it is similar to dysplasia in benign tumors, but is much more extreme. Now the term anaplasia literally means to grow backwards, and this reflects the fact that at one time tumor cells were believed to undergo a regression with regard to differentiation. However, present dogma is that most tumors arise from stem cells that may or may not continue to undergo maturation. Thus, according to this notion, well-differentiated tumors still undergo differentiation, while anaplastic ones do not. Also note that some malignant tumors also show the property of pleomorphism, which is a variation of shape and size. This property applies to the cell as a whole, as well as to the nuclei, which may vary with regard to their size, their shape, and their staining density. Now at this point, I would ask you to move on to part 2 of this video series.